Bismillahirrahmanirrahim. Let's start today's lecture. So our today's lecture is very important, and that is titled as Microorganisms of Industrial Importance and Their Sources. So whenever we are in a trash, whenever we have nothing in our hand in order to produce something in industrial biotechnology, so what is the fundamental requirement that we need in order to produce the bioactive compounds is actually the microorganisms. So we need those microorganisms that are known to produce some antibiotics, some enzymes, or whatever the compounds that we need in industrial biotechnology. So today we are going to discuss what are the sources of those microorganisms, from where these microorganisms could be obtained, and they could be grown in order to produce various industrial bioproducts. So let's start discussing the sources of microorganisms. So very first and fundamental source of microorganisms, when you know nothing about the microorganisms in order to produce the industrial products, is literature. So when we know nothing, then literature is our best companion. So we can read literature, we can go through literature in order to find those microorganisms that are known to produce the required industrial products. So whenever we see the literature, the things that comes in our mind, that should come in our mind, is the research papers. So going through the research papers is the fundamental requirement in order to obtain knowledge, in order to know about the industrial microorganisms that are known to produce particular bioproducts. So until or unless we do not go through the literature, then we could not be able to find any microorganism or any industrial process that that is needed to produce the required industrial product. So in literature, research papers are the fundamental requirements. Research papers that belong to particular microorganisms, that belong to particular industrial processes, that belong to particular uh, patented processes that are needed in order to produce the industrial products. So going through these research papers is the fundamental aspect and the first requirement, first point of start in order to produce the industrial products, especially in the cases when we do not know anything about the industrial microorganisms. Then another source that solely belongs to others, and that belongs to literature, so is whenever we go through the literature, whenever we go through the patented processes, whenever we go through the research papers in order to find the information about the industrial microorganisms. So uh, what we could know about is the microorganisms that have a particular number. For example, the microorganism could have a particular number of a particular culture collection. So this means that we should also know about the international culture collections that are responsible to store the microorganisms of industrial importance. So going through the literature, we should also know about the international culture collections. And while knowing about these international culture collections, what benefit we could obtain is we can request these international culture collections in order to send a particular microorganisms for the synthesis of a particular industrial product. So in international culture collections, there are so many international culture collections that exist. Uh, for example, ATCC, and you might have know about the fact that whenever you go through the literature, you find microorganisms that are that are represented with a particular ATCC number. So this ATCC number is actually belongs to the international culture collection where this particular microorganism has been stored. So ATCC stands for that stands for uh, type strain, international type strain culture collection. Sorry, I don't know about A, type strain culture collection. So the second culture collection that is very famous is NCBI, that is National Collection of Industrial Bacteria of Britain. National Culture Collection of Industrial Bacteria of Britain. So that is the second famous culture collection. So this means while we are starting from a start, we should go through the literature, and at the same time, we should also know about the internationally famous culture collections from where we can obtain the microorganisms in order to start our industrial processes. Then, at the second point, if we do not go to the literature, if we don't want to go to the literature, and we are trying to start from our own microorganisms, then what we need is, in industrial biotechnology, is to isolate the microorganisms, right? So one aspect is to go through the literature and find the internationally known microorganisms, and then obtain those microorganisms from internationally known or internationally famous culture collections. And the second method is to isolate our own microorganisms. So this is the second strategy. So if we adopt the second strategy and we isolate the microorganisms in our own laboratory, then we can apply various technologies. We can apply various techniques in order to isolate the microorganisms in our own laboratory. And you might have we have learned so many times about these technologies. We have learned so many times about these techniques. And the first one is the serial dilution method. So this is very well known method of the isolation of microorganisms. So serial dilution method is actually isolating the microorganisms from the rich sources of the environment, right? Serial dilution means we are diluting the sources from where the microorganisms are going to be isolated. So from where these microorganisms could be isolated, there are so many sources, there are so many media there are so many natural medias from where these microorganisms could be isolated. For example, we can isolate microorganisms from soil, from water, from marine environment, from harsh environment, from litter, from tree trunks, and so many other. There are so many other ecosystems. There are so many other environments or sources from where these microorganisms could be isolated. So serial dilution method is the fundamental method in order to isolate any particular microorganism to be used in industrial biotechnology to produce various industrial products. So in serial dilution method, if you see a picture here, if you look at the picture here, so you see what we do is we collect a particular sample from a particular source. Let's say we can collect samples from water, or we could collect sample from soil. So in the next step, what we could do is we would perform a serial dilution. So one thing, we can also directly spread this water or the soil directly onto the Petri plates in order to grow the microorganisms. But the reason not adopting this method of directly inoculating the soil or water directly onto the Petri plates is that the environment from where we are collecting the microorganism is so rich in the density of microorganisms that whenever we grow directly these microorganisms to the Petri plates, what happens that we would ultimately find a smear. We would find so many microorganisms growing on the Petri plates, and we would not be able to handle these microorganisms independently. We would not be able to distinguish these microorganisms independently. So what we do is we perform a serial dilution then. Serial dilution mean we dilute our sample of an environment. For example, if we have a water or if we have a soil, we are going to perform the dilution of this sample in various steps. So what we do is, in the first step, we add a portion, let's say 1 gram of our soil into the first test tube. Then we mix it, and then we take 1 ml from this test tube and we add this 1 ml to the next 9 ml of the second test tube, and we repeat this process in so many number of test tubes, and this process is then called as serial dilution method. So the fundamental aspect is our purpose is to serially dilute the rich environmental source so that we could dilute the density of the microorganisms in various test tubes, so that ultimately in the last test tube, which is the most diluted one, whenever we take the sample from the last test tube and we grow on the petri dish, then we could be able to find the microorganisms that could be distinguished and that are separated on the petri dish. So this is the fundamental reason why we perform the serial dilution instead of using the direct rich samples for growth purposes. So the second step, the second strategy, one strategy is the serial dilution, and the second strategy is the direct selection. So direct selection means we do not perform the serial dilution. We directly select the microorganisms from the richer source of the environment, right? So direct selection means we directly inoculate our sample from the environment directly to the media without performing any serial dilution. So in direct selection, one thing that that should be kept in mind, that in direct selection we are interested in a particular category of microorganisms, and we do not care about the other categories of the microorganisms. So suppose there are so many groups of the microorganisms in soil sample, there might be so many groups of the microorganisms present in the water sample. So we are particularly interested in a specific category of the microorganisms, and we want to kill the other categories of the microorganisms. So uh, let's suppose we are selecting a particular category. So what we do is we add a particular substrate on the petri dish. Let's suppose we are going to select the alpha amylase producing microorganisms, right? This is an enzyme. So if we are interested in alpha amylase producing microorganisms, what we do is we would add in the agar media the starch. So starch is the substrate where alpha amylase producers would grow, and they would produce a particular zone there. So we can select the alpha amylase producing bacteria out of the huge density of the other microorganisms that might be present in the sample. So what we do is we add the starch and we directly inoculate our soil or maybe the water onto the petri dish. And what the alpha amylase producers would do, that they would produce a particular zone on the petri dish. And in this way, out of the millions of the bacteria, out of the billions of the microorganisms that would be present in the environmental sample, we could be able to distinguish only the alpha amylase producing microorganisms. So this is called as direct selection of the desired microorganisms on the agar media, right? And then the third and last uh, technology, the third and last technique that we use in order to find the microorganisms of industrial importance is the enrichment of the microorganisms, right? So enrichment is totally different from whatever we have already studied in case of serial dilution, or from whatever we have already discussed in case of the direct selection of microorganisms. So in enrichment of the microorganisms, we apply some of the sub techniques, like we could incubate our soil to the substrate, we could enrich our microorganisms to the liquid media, and we can also enrich our microorganisms to the toxic analog of the substrate. So let's discuss one by one about these techniques. So first of all, we should know about the enrichment. What is the enrichment? Enrichment means that our soil sample or our water sample that we have already collected from the environment is rich in microorganisms, right? It has huge number, diversity, and variety of microorganisms that could be present in a particular sample of the environment. Then what we are going to do is we are now interested in a particular category of microorganisms. This is exactly the same case as we have already discussed in case of selection. In selection, in case of selection, we are also interested in a particular category. But in selection, we directly select the microorganisms on the agar media. But what we do in case of enrichment, we are first interested in increasing the number of those desired category of microorganisms in the sample, and then we select those microorganisms. So enrichment means increasing the number of the particular category of microorganism by using an artificial media or by using a particular substrate in the soil or in an artificial media, and then selecting that particular category of microorganisms out of the variety of the other microorganisms that might be present in the environmental sample. So in the first technique, what we do is we incubate our soil. For example, if we are going to enrich our particular category of microorganisms from soil, so we must have information about the substrate of a particular category that we are interested in, right? For example, if we are interested in a particular category of microorganisms, that we must know about the substrate on which that particular category of microorganisms could grow, right? Then what we do is we add that substrate directly to the soil from where we are going to take the samples in the next stage. So whenever we add the substrate directly into the soil, in order to increase the number of that particular category, what would ultimately happen that we would add the substrate, then what happens that the number of that desired category of microorganisms would start increasing, right? The number would start increasing. And with the passage of time or with the incubation time, what would ultimately happens that the number or the density of that particular category of microorganisms would be increased many folds, and the density of the other microorganisms would be reduced. So it means that our desired microorganisms are now enriched in the soil. So this would happen ultimately due to the addition of the substrate in the soil. So we would add the substrate and then we give a particular incubation time. And after that incubation time, what we do is we collect the sample from that soil and we directly culture these microorganisms onto the agar media. So then what would happen that because of the enrichment procedure, the soil would now contain, there are maximum chances that the soil would now contain only that particular category of microorganisms in which we are interested in. So this is the incubation of soil with a particular substrate. And then the second type of the enrichment is the enrichment in the liquid media. So you see in the first step that we do not use the liquid media in the first step, right? We are enriching our particular category of microorganism directly in soil, and then we are taking these microorganisms onto the artificial media in order to grow them and in order to use them for various industrial procedures. While in case of the second type, what we are now going to do is we are now enriching our desired category of microorganisms in a liquid media rather than directly onto the soil or rather than directly onto the agar media. Now we are using the liquid media. So if we are using the liquid media in enriching our desired microorganisms among various other microorganisms that might be present in the media, in the environment. So what we do is we then add a particular substrate in the flask in the liquid media, right? So the category in which we are interested in, we should know the specific substrate of that category. And then we add that particular substrate into the liquid media. and then we inoculate our sample to this particular flask. So what would happen that when we grow this particular flask uh, by shaking, so what would happen that in the second step, only that particular category of microorganisms which is growing on that particular substrate would, its number would increase, its population density would increase, while the population density of the other microorganisms would be reduced. So then we perform another step. We take up a specific amount of inoculum from this flask and we add this into the fresh media. So in the second step, what would happen that there might be the greater chances, the chances of increasing the number of that desired microorganisms would be increased many folds. So in the second step, our desired category of microorganisms would be in much greater number as compared to the other microorganisms. And in similar fashion, we perform these steps in number of other time. For example, we take a particular inoculum, particular amount of inoculum from this flask, and we could add this inoculum to another flask in order to grow them and in order to increase the number of our desired microorganism. So every time when we are enriching and when we are repeating this step, every time we are enriching the number of our desired category of microorganism. So this is the enrichment in the liquid culture. So ultimately, after repeating so many steps, what would ultimately happen that only the desired category of microorganisms would survive and other microorganisms would be removed or their number would be decreased many folds in the flask. So then we can grow these microorganisms to the agar media and we can use these microorganisms for various industrial applications. Then another step, another very interesting step in order to enrich the microorganism is the enrichment with the toxic analog of the substrate. So this is very interesting to understand. Rather than adding, uh, 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 either we are using the direct, either we are enriching our desired category of microorganisms directly in the soil, or we are using artificial media, or we are using the liquid media. So what we do is, in the third step, what we do is, uh, uh, in this technique, rather than adding the substrate, we add the toxic analog of the substrate. Toxic analog means that the molecule whose chemical structure would be very much similar to the substrate, to the original substrate, but it is toxic to the microorganisms, right? So there are some chemicals that could be chemically analog to the original substrate, but their effect is toxic. So we would then add the toxic analog of the substrate directly to the soil or in the flask. So what would happen that whenever we add the toxic analog and we inoculate the environmental sample into the flask, the desired category of the microorganism would start dying because the analog is toxic, right? So the desired category microorganisms would start dying. So there are chances whenever we repeat this process or whenever we incubate our soil with the toxic analog, so there are chances that some of the microorganism that belongs to the desired category of the microorganisms would survive. And if they are survived, their genome has been, there are chances that their genome has mutated and it has developed resistance against that toxic analog. So what would happen that particular microorganisms who has now survived against the application of this toxic analog is now would be more productive as compared to the original category of the desired microorganisms. So if we could isolate those microorganisms that are surviving by applying the toxic analogs of the substrate, so it is thought that those microorganisms would then be more productive industrially as compared to the original microorganisms of the desired category. So this is all about the sources of industrial microorganisms, that how we could isolate, how we could enrich them, and how we could obtain the already known microorganisms that are elaborated in literature from various culture collections. So thank you very much. This is all about today's lecture. Now, if you have any question, you can ask.